When you're ready.
Today we're going to be talking about gene editing. And we're going to be looking at it from the historical, the scientific, the ethical, and the futuristic perspectives. Throughout the decades, gene editing has evolved a tremendous amount. Starting in the mid to late 1900s, it was designed to change the DNA in an organism such as plants, animals, and bacteria. With the main focus on modifying genomes in a living organism, this idea was used to help improve the understanding of chromosome function and evolve ways in which it treats genetic and acquired diseases. So first I'm looking at the history. Looking back at the mid-20th century, the production of genetic mutations were studied. During this, it was found that genetic mutations could be enhanced with chemical treatments or radiation. Before editing, the idea of identifying DNA first came about in 1869, in which Frederick Mischer, a Swiss doctor, became the first to identify DNA as a molecule. By the 1980s, genetic engineering was evolving. This then brought in real treatments and vaccines in 1981. Thomas Wagner and his team were able to transfer the gene of a rabbit into a mouse gene by using the idea now known as DNA microinjection. This helped to later evolve revolutionary transgenic animal experiments. Later in 1987, the idea of CRISPR was started, but the actual idea was not put into practice until 2012.
Next, I'll be speaking from the scientific lens. Various technologies have been implemented to help with gene editing, such as zinc finger nucleases, talons, and meganucleases. The most recent and popular gene editing tool is known as the CRISPR-Cas9. It's the most powerful tool for understanding how genes work. Since CRISPR-Cas9 is an RNA-based system, it can be altered more quickly and easily than plant-based techniques and can target many locations. When using CRISPR-Cas9, a DNA sequence is cut at a specific genetic location, and the DNA sequences are either deleted or inserted. This process can change a single base pair of DNA, or the significant portions of chromosomes, or the regulation of gene expression levels. CRISPR offers another treatment option for patients not responding to traditional or first-line therapies. CRISPR-Cas9 gene technology is being used to find ways to treat diseases such as inherited eye disease, Alzheimer's, and Huntington's disease, as well as non-inherited diseases such as HIV and cancer. Gene editing can also be used for treating and reducing two significant infectious diseases that we all know of, COVID-19 and influenza. Researchers at Stanford University have improved a gene editing tool known as the CRISPR-Cas13a to split and destroy the genetic code of the virus responsible for COVID-19 in order to prevent it from infecting lung cells. This technique, known as the Pac-Man method, can significantly decrease the amount of virus in the solution more than 90%. The Pac-Man method can play a significant role in minimizing morbidity and mortality rates in COVID-19. A research group at Georgia Institute of Technology had adopted a similar approach of gene editing technologies that can treat mice infected with influenza. Researchers state that the treatment for influenza in the process can be effective against 99% of all influenza strains. Having another treatment option for these diseases will reduce the burdens of illnesses, medical visits, hospitalizations, and increase the quality of life for humans.
Now I'm going to be going through the ethical lens. With gene editing in the medical field, there's different forms of these procedures that can take place. One is somatic, where scientists are going to be editing on disease-causing DNA that's within non-reproductive cells. But unlike this, germline procedures can harm the sperm and the eggs, and along with this, these possible effects could go into future generations. But within the somatic versus germline discussion, another issue is improper consent. Since these procedures are done on embryos, the patient would not be able to give their consent. And since these procedures are very serious and they can cause harm to the future generations, and the success isn't guaranteed, any patient should be able to contribute to that decision-making process. Another controversy within gene editing is the possibility of enhancements, meaning changing physical traits. A 2016 Pew study of surveys had said that 4,000 individuals revealed that they had anxiety outpacing enthusiasm towards not only enhancements with somatic editing, but also with mechanical and transplant-related enhancements. And along with this, we're also getting rid of a standard in society. And by this, what I mean is we would no longer be able to use words like good or bad to describe someone's abilities because we've all been genetically modified to be the same. Lastly, I'm going to be going through the Catholic Church's viewpoint. So the Catholic Church is against gene editing in the ways where it is dishonoring God's image and is also going against the dignity of the human person. So with procedures like germline, where it can affect future generations, it is thought that we're also changing and affecting God's image for those future generations. Acts of the Apostles chapter 17 verse 29 also says that, since therefore we are the offspring of God, we ought to not think that the divinity is fashioned from gold, silver, or stone, but from human art and imagination. And this means that we are more than just the genes that we do contain. We're also not respecting individuality because we can start to create stereotypes and judgments towards people's genetics that they have gotten and the traits that they do obtain. Lastly, we're not respecting the free will that God has given us. He's given us this free will, and the germline procedures, since the patients are the embryos, we're not upholding that gift that God has given us.
Lastly, I'm going to be going through the futuristic lens. Genetic modification is showing immense growth and it has a very great future promise of success. The University of Pennsylvania has used CRISPR on cancer genes. They conducted an experiment that explored an immunotherapy technique that alters a patient's immune cells to more effectively detect and destroy cancer cells. This was seen as a proof of principle, feasibility, and safety by Dr. Stottmeyer. He also said that this now opens up a whole world of CRISPR. Technological advancements like these are taking the first steps to transform and improve humanity. Victoria Gray is another patient who has used CRISPR to treat her sickle cell anemia. The results of her trial have been amazing for the past couple of years, but the problem is scientists and doctors don't know the long-term effectiveness and safety. This leads into some safety drawbacks when it comes to these trials and technologies. In terms of the future in these therapies, if they're going to be available to all patients, research is going to need extensive safety research. This research is inevitable, but it's needed to prevent harm. Once these trials are proven 100% safe, they can then be applied and used in more patients in the future. Another great thing about CRISPR is how researcher Jerry Lee says that the convenience and technique of CRISPR is becoming mainstream. It's just going to take some time to get used to the strengths and risks of this technology. Genetic modification is a bright future ahead once it's passed through the lengthy safety procedures. Then research will grow and it'll be made available to even more patients. Through these lenses, we've determined a solution that gene editing should be limited to just medical use and not physical enhancement. The pros of this solution is that by removing the cells from the patient and modifying them, it will be made possible to prevent hereditary diseases, treat them in real time, and to give some relief to those suffering from incurable diseases.
For the cons, the first one that we have up there is hard to stop. And what we mean by this is, as that research continues and the possibilities and the success rate increases, it'd be difficult to prevent people from going through with these physical enhancement procedures. And even if they created laws to stop that, that could anger a lot of people in our society. And then the safety risk and unknown effects kind of goes hand in hand, and this can go specifically with that germline editing, where it can first of all affect the future generations, and still as of right now, as the research hasn't gone to its full potential, the risks aren't fully known, and it can come with unpredictable harm.
Thank you. Big round of applause. Come on, huge round of applause. All right, a couple of questions. Give one specific way your thinking changed as a result of learning about Erin's findings.
My thinking's changed after learning about Erin's findings because I was unaware of how much the topic had evolved over time. And I didn't have the understandings of how much science had been put into the topic about how CRISPR was far from the start of genome editing.
Okay, thank you. Erin, what is a way in which your team's resolution makes you think differently about your own individual research?
I try to base most of my research off my team's resolution, but what makes me think differently about my own research is how I focus on how gene editing started and where it is going, as where our conclusion shares on how genome editing should be used for medical purposes and not physical. Although I do talk about somatic versus germline in my paper, I do not talk about how one is seen as ethical and the other is not.
Okay, thank you. If you had another team member, Brooklyn, what other perspectives or limitations could they have researched and that would have made a useful contribution to your project?
I think that we would include an economic lens because as I did the ethical, it's not fully really statistics, but more just like viewpoints that have been obtained from different groups within our society. But I think that economics could really help to bring in statistics into it and help gain whether it is right to do it medically or how much the cost goes into it. And especially if it is going to be a great possibility in the future and really cure and helping cure and prevent diseases, how much money would it go into it and if that would be beneficial.
Okay, thank you. And finally, Sloane, what is an example of a compelling argument from one of your individual reports that you decided to exclude and why?
Well, when reading Ashley's individual report, I found a compelling argument about gene editing techniques using zinc finger nucleases, which are DNA-binding proteins that have targeted editing of the gene, creating double-strand breaks in DNA at specific locations. We excluded some of these specific things like this because we felt that it was sort of unnecessary to the main idea, and with a given amount of time in the presentation, we couldn't put it all in.
Okay, thank you.